Inside pigs, embryonic humanized kidneys were grown for a period of 28 days by researchers. In a remarkable breakthrough, researchers at Guangzhou Institutes of Biomedicine and Health have achieved the creation of chimeric embryos comprising of pig and human cells. The scientists managed to transfer these embryos into surrogate pig mothers where they produce striking results: development of fully functional human-like kidneys with normal tubule formation and architecture within 28 days. While past studies have shown success in generating human tissues like skeletal muscle or blood, this was the first time that a complete and fully functioning humanized organ was grown inside another species. These exciting findings were recently published on September 7th in Cell Stem Cell. Focusing on the kidneys, the researchers studied these organs as they develop early on, and because they are the most commonly transplanted organ in human medicine. Pigs have been unsuccessful hosts for growing human organs in previous attempts, whereas mice have been used to produce rat organs and rats have produced mouse organs, explains Liang Shui, lead researcher from the Guangzhou Institutes of Biomedicine and Health at the Chinese Academy of Sciences and Wuyi University. Through our innovative method, we are able to enhance the integration of human cells into host tissues, ultimately leading to the successful growth of human organs in pigs. Overcoming the innate incompatibility between pig and human cells while deploying human stem cells into pig embryos has arisen as quite a task. At the Guangzhou Institutes of Biomedicine and Health, senior author Guangjin Pan has been applying their efforts to surmounting this feat. Our research has focused on bridging the gap in interspecies chimera efficiency, mentions Pan. By pinpointing certain vital factors that promote healthy cell competition, we have made significant strides towards achieving our goals. Dependent on three critical constituents is the team's method. Using CRISPR, scientists removed two genes necessary for kidney development from a single-cell pig embryo, allowing it to develop with a unique niche for the introduction of human cells without competition from pig cells. Making human pluripotent stem cells more adaptable and less likely to self-destruct, researchers engineered the cells after temporarily halting apoptosis. These newly engineered cells were then transformed into naive cells, resembling early human embryonic cells, through special medium cultivation techniques. Growing the chimeras in optimal conditions for nourishing both human and pig cells happened before implanting the developing embryos into surrogate sows. Since these cells typically have different requirements, unique nutrients and signals were necessary. Thirdly, this was part of the process. Assessing if humanized kidneys were successfully produced, the researchers implanted 1,820 embryos into 13 different surrogate mothers. They ultimately ended gestation after 25 or 28 days before extracting the embryos. For the study, 5 hybrid embryos were analyzed, 2 at 25 days and 3 at 28 days after implantation. Remarkably, these embryos demonstrated kidneys that were both structurally normal and contained between 50–60% human cells. During examination, it was observed that the kidneys were in the mesonephros stage of development and had already begun forming cells that would become the ureters later on. This is the second stage of kidney development, and the formation of tubules and buds was noted. Throughout the embryos, the team examined the possibility of human cells contributing to other tissues. This discovery could have ethical consequences if a lot of human cells were discovered in neural or germline tissues and the pigs were brought to term. They discovered that most human cells were situated in the kidneys, while the rest of the embryo was made up of pig cells. According to Zhen Dai, a senior author from Guangzhou Institutes of Biomedicine and Health, we discovered that by carving out a spot in the pig embryo, the human cells will naturally fill those gaps. Our observations revealed minimal human neural cells in the spinal cord and brain, and no human cells around the reproductive area, leading us to believe the human pluripotent stem cells did not transform into germ cells. The researchers speculate that by disabling more genes in the human pluripotent stem cells, this outcome can be averted, and this theory can be examined in later investigations. The team is presently concentrating on growing human organs in pigs. They have successfully optimized growing humanized kidneys using human-pig chimeras. However, the team is now planning to allow the kidneys to develop for a longer duration. In addition, they are also attempting to produce the heart and pancreas in pigs. The researchers understand that achieving the ultimate objective of perfecting this technology for transplanting human organs will be a challenging and time-consuming endeavor. Successfully cultivating a fully operational human-like organ in a swine would entail supplementary measures due to the fact that organs are made up of numerous separate cells and tissues. To create an isolated environment for just one particular type of cells, the researchers established a niche that resulted in the kidneys containing vascular cells taken from a pig, which could cause the organ to be ineffectual in a transplantation. The process of creating an organ where everything is comprised of human cells is not a simple one, as organs are typically made up of multiple types of cells. Miguel A. Esteban, senior author at Guangzhou Institutes of Biomedicine and Health, explains that engineering pigs to achieve this would require a more intricate approach, as well as additional obstacles to overcome. To analyze the formation of human organs and developmental illnesses, this method can be utilized in the interim. Esteban explains that delving into the late stages of developing organs that can be used in clinical settings isn't the current focus. Instead, this process allows for the examination of human development. By tracking the infused human cells and their manipulation, ailments and cell lineage formation can be studied. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.